Can you guys can you see the presentation, the
Yep,
slide?
looks great.
Okay, awesome. Well, thanks everyone. My name is Renato Bergeri, and today I'll be sharing with you a bit of the work that I've been developing in the past 3.5 years or so, since I started my postdoc at JPL, and now I'm a research scientist at Caltech, JPL affiliate. And this presentation is really this connection between nutrient cycle and carbon. And when we think about climate changes, we don't usually think about roots. But in this talk, hopefully by the end of it, we'll be thinking about climate change and how roots and the rhizosphere can interact with climate change. I want to thank my collaborators here. I'm not gonna read all the names on the list, but these guys are awesome and they are all over the world. So thanks to them as well. So let's dive into that. So first of all, the land surface is a large carbon sink. It absorbs almost 1/3 of all the anthropogenic CO2 that we put up in the atmosphere. So here on the left-hand side, I show the CO2 flux, the CO2 emissions since pre-industrial times. And so the gray area is representing the fossil fuel emission of CO2. And the yellow area represents the land use change emissions. And in the negative part of this graph, we can see three colors. And so when the CO2 is emitted, the CO2 goes into three main compartments of the Earth system. The dark green area represents the ocean sink. And we can see how the ocean sink is quite smooth along the years. So the more CO2 we add into the atmosphere, the more CO2 the oceans uptake. The light green part of the graph shows the land sink. And we can see how the land sink uptake is quite bumpy year after year. So for some years, the land is acting as a very strong carbon sink, and some years not that strong. The remaining part, whatever is now going into the oceans or being absorbed by the land surface via photosynthesis, remains in the atmosphere and causes climate change. The figure on the right-hand side shows a time cut of the same plot, but now the sink is the positive axis. And we can see that since the 60s, the land surface has been acting as this relatively strong carbon sink, mainly due to the CO2 fertilization effect of photosynthesis. And so a way to estimate the land sink is running land surface models. Land surface models are the land compartments of climate models, the Earth system models. And we can see how individual members, represented by the light green lines, disagree substantially among themselves, not only in the sign, but also the magnitude of the land uptake. However, when we take the ensemble member or the mean of all these land surface models, we get the dark green line. And the dark green line kind of agrees quite well with the residual budget. And the residual budget is calculated as whatever is now remaining in the atmosphere or going to the oceans. We blame the land surface because most of the internal variability and the uncertainty of the global carbon sink is due to the land. And so along the years, we can see that these land surface models, or at least the ensemble member, are doing a great job, well, a relatively good job in representing the land sink globally. But well, we as the Earth system scientists want to know if the land surface will keep acting as a strong carbon sink at the end of the century, because our political agenda depends on it. Let's suppose that from nowhere, the land surface stops absorbing all that carbon, and so climate change will just speed up and things will go south very quickly. So a way to calculate if the land surface will remain a carbon sink by the end of the century is running these Earth system models. And so the CMIP-5 simulations is this exercise where all the most famous groups, climate modeling groups in the world, get together and perform these simulations in the future with their ensemble, with their individual members. And then we can get the ensemble member of all these climate models and see what is going to be the change in NPP. NPP is net primary production. So it's photosynthesis minus autotrophic respiration. Autotrophic respiration is the carbon that plants respire to support metabolic rates, and etc. And so when we look at the change in net primary production between the end of this century and pre-industrial times, we see that NPP is going up, represented here by this black line. And of course, with a lot of uncertainty, not only due to the different scenarios of climate change, of CO2 emission, but also because these individual models represent different things. And so these figures here were taken from this paper published in Nature Geoscience in 2015, where the authors asked the question, okay, so these Earth system models are saying that because of the CO2 fertilization effect, net primary production is going to be around 40 petagrams of carbon per year extra in comparison to pre-industrial times. Now, what would be the amount of nitrogen and phosphorus, the two main nutrients, required to support that initial photosynthesis predicted by those models? And when the authors calculate the amount of available nitrogen and phosphorus globally, they reveal that number downwards. So when they add a nitrogen limitation, they decrease the initial prediction in 19%. And when they added phosphorus on top of that, the initial prediction goes down 25%, which in turn will turn the land surface into a carbon source by the end of the century, and not a carbon sink, here represented by these negative values. And so, is that true? Well, in the past few years, we have been seeing more evidence suggesting that nitrogen and phosphorus do constrain the CO2 fertilization effect and global plant biomass. And phosphorus is particularly important for tropical forests. Tropical forests are the hot spots of productivity of global photosynthesis, and tropical soils are relatively poor in phosphorus. So we have some in situ evidence suggesting that phosphorus does limit photosynthesis. Now, we still don't have any consensus over that question. Sorry, I can hear some feedback. Is that a question or just an open mic?
No, I think someone was unmuted briefly.
Okay, all right. So after that paper was published in 2015, a group of researchers wrote a reply saying that the land is unlikely to become a large carbon source, mainly because there are several feedback processes that were not considered in the initial exercise about, using the bookkeeping methods to calculate the amount of nitrogen and phosphorus readily available for plant uptake. And these feedback processes include mineralization of nutrients from soil organic matter and processes governing the recycling and uptake of nutrients. And so it is still an open question. Now, what are some of these missing feedback processes? Well, for years, humans have been thinking about trees communicating with us or other living forms on Earth. And this paper in 2010 in New Phytologist found that in a patch of Canadian forest, one tree was connected to other 47 trees via this fungal network or this mycorrhizae in the soil. And so what is mycorrhizae? Mycorrhizae is the connection between roots, the root system of a plant, and fungi. And it is a symbiotic relationship where plants pay out carbon to the fungi, and the fungi eats that carbon and in turn goes around the soil, scavenging the soil for nutrients. So it is a win-win situation. And again, Earth system models do not represent that type of association. And so now it is known that plants don't get nutrients for free. You know, acquiring nutrients is an expensive process, and plants need nutrients to support photosynthesis. And so, coming with the idea that plants do not get nutrients for free, can we use then a resource optimization theory to calculate nutrient uptake in these climate models? And this is what the concept of the FUN model is. So FUN it stands for fixation and uptake of nutrients. And so the idea of the FUN model is that after we calculate net primary production, that means photosynthesis minus autotrophic respiration, before we allocate that carbon to plant biomass or to plant growth, we will use part of that carbon to acquire nitrogen and phosphorus. And we will do that via multiple uptake pathways. So here I just name a few. So in the model, we can calculate, for example, retranslocation of nutrients from leaves. In the case of nitrogen, we can calculate biological nitrogen fixation from the atmosphere. We can calculate the two different mycorrhizal types, so arbuscular mycorrhizae and ectomycorrhizae from soils, and also the direct root uptake or the non-mycorrhizal uptake, where roots are in direct contact with that soil. And basically what we do is we calculate this cost function. And so we have here N, and I promise this is the only equation that I'm going to show in the entire presentation. Here NS mean is the amount of nutrient in that pool, and the pool can be leaf or soil. And C root is the amount of root biomass in the soil. And so we calculate this cost parameters K associated with each and every one of these pathways. And what we do is we know that plants want to save resources like we do. And so they will always choose the cheapest uptake pathway and acquire nutrients via that uptake pathway. And so how can we find these cost parameters? Well, we use some feeding cost functions to find these parameters. So the first paper that was published about this model was in 2010 in GCB, where we had a bunch of sites here, some phased sites. Phase is a fertilization experiments with CO2, and other sites on Earth where we had observed nitrogen uptake, and we fit those K parameters to those different uptake pathways. And so the model was expanded since 2010. So in 2010, the model only represented nitrogen, and it makes sense to start for nitrogen because nitrogen is the most limiting nutrient that we can think of. The concentrations of nitrogen in the plant is much higher than everything else. Nitrogen is available in rubisco, which is used for photosynthesis. So the model was first developed for nitrogen, but after that, the model was expanded for mycorrhizal uptake as well. And more recently, we mimic the nitrogen uptakes to phosphorus uptake and found the same cost functions. So the FUN model, which was developed for in situ, with in situ data, was then coupled into a climate model, into the community land model, the land surface model of NCAR's Earth system model. And the beauty of doing
sorry,
this,
Renato, can I ask a question here?
Sure.
So maybe I'm not fully understanding how the FUN model works, but is the implication here that the data you're fitting to was able to kind of isolate each of these individual potential paths? Or are you saying that actually it was just kind of a homogeneous thing and you're kind of like making an assumption that these are the paths and the real biological system is kind of taking the path of least resistance, so to say? Like when you're fitting, for example, and parameterizing the costs of a specific branch, like getting nitrogen specifically from arbuscular mycorrhiza, is that involve fitting data from like a very targeted arbuscular mycorrhizal experiment, or is it kind of from a broader thing and you draw inference?
From a broader thing. So it's I'm going to talk about that a little later, but we will see that data is very rare in that realm, right? Like it's very rare to have people actually measuring what is the nutrient coming from mycorrhizae. So what we have is the general uptake. So we have an observation of the total uptake. And what we do is that we're going to do exactly as you described. We're going to assume that the plants will always choose the cheapest one. And so to acquire that result, we're going to fit the parameters that will give through the cheapest one.
I see. So the kind of information you have to work with is like you have the total fluxes, and also you have maybe like measures of like how much mycorrhizal there are or how much,
Yeah,
and then you kind of draw. Okay, I see. Okay,
exactly.
thank you.
Yeah, and I think that's going to be a little clear as we go along. But yeah, thanks for your question. And please feel free to stop me if you have more. So when we couple this model into a nervous system model, we actually can get now these global maps. And these global maps were quite rare. I mean, it's very rare to find a global map of mycorrhizal uptake versus direct root uptake. And so now it's interesting to see these global patterns here. And it's kind of interesting to see how a model that was developed for in-situ data can actually show, for example, the nitrogen fixation peaking in the savannas, which is something that we know that happens and we observe in real life. So a model that was developed for just a few sites, 15 or 16 sites, can appropriately predict that symbiotic fixation peaks in the right places. So we were quite confident. Now, there's a lot of uncertainty in these maps, of course. But what about phosphorus? And so we kind of like developed the same idea for phosphorus now, and we added the fun model into ELM. CLM, the community land model, doesn't have a phosphorus cycle in it. And so we need to find a nervous system model that had a representation of the phosphorus cycle. Things like immobilization and mineralization of phosphorus and nitrogen from soil organic matter, things involved, fluxes involved in the phosphorus cycle, so sorption and desorption of phosphorus from occluded sources, weathering, nitrogen phosphorus deposition, litter fall, vertical transport, and horizontal transport. And all I did was to now add this concept of resource optimization with the different uptake pathways. So now we have ectomycorrhiza, arbuscular mycorrhiza, symbiotic fixation, the direct root uptake, and retranslocation from leaves. First, before we move on into the actual results of this model, we had to do some validation. Every time we add a new function in a nervous system model, we need to be very careful to see if we're not going to break anything that is already working properly in that model. And so we use an international LAM model benchmarking tool, ILAM, that includes 16 global data products, including flux tower data, satellite data, machine learning techniques. And we looked into those variables. So biomass, global photosynthesis, leaf area index, ecosystem respiration, soil carbon, and for the hydrology cycle, we look into evapotranspiration, and also how these different variables associate with climate data. So what are the relationships of these variables with precipitation, temperature, and radiation, incident radiation. And we have a relative score, and this relative score accounts for bias between the variable and the observation, account for root mean square error, account for the timing of the peak of the seasonal cycle of these variables. And here E3SM is the original model, and E3SM Fund 2 is the original model with nitrogen only, and Fund 3 with nitrogen and phosphorus. And this relative scale shows that the purple colors indicate better agreement with, between the model and the observations. And so when we were surprised that when we added the fun model with nitrogen and phosphorus, we actually improved all the agreements of the Earth system model with those data sets globally. And so now we can have the same sort of maps for phosphorus uptake. So we can have here an estimate of what is the global phosphorus uptake via mycorrhizae. And so this number here appear from the model, 20 teragrams of phosphorus per year, where most of the phosphorus comes from our arbuscular mycorrhizae located in the tropics, and most of the extratropics are associated with ectomycorrhizae, and most of the phosphorus comes from that area. And when we compare nitrogen and phosphorus uptake, and here is the zonal plot. So here is the average in latitude. So we can see how the equator, where the tropical forests are, shows most of the total annual nitrogen and phosphorus uptake. But it's interesting to see how different uptake pathways have different weights in acquiring phosphorus and nitrogen. So for example, the red area show ectomycorrhizae and the blue area arbuscular mycorrhizae. And we can see how mycorrhizal associations are very important for nitrogen uptake. Now for phosphorus, we see how direct root uptake, the pathways have kind of a split importance for phosphorus. As you may know, phosphorus recycles in geological timescales. So for a plant, it's much more economic to retranslocate phosphorus than acquire new phosphorus that is quite rare. And so that was an interesting result here, because you often think that mycorrhizae is responsible for 80% of nutrient uptake, but that is true for nitrogen, but it doesn't seem to be true for phosphorus. And so when we go back,
can I ask a quick question before we move on? 2 slides back, you showed us this improved performance with various metrics. So I assume that the improved performance comes from, I imagine, sort of 2 things. One is that your model now accounts for like natural phenomena that aren't in the older models. And then the other is that you have some data that goes into the model now that didn't go in, like you know something about where the ectomycorrhizae are or something like that. Like, could you just run through for me so I understand like what additional data goes in here that is, and how available it is and where it comes from and stuff?
Sure, I think I will touch that in a minute.
Okay,
right
wonderful.
after that. Yeah.
Okay, great.
And yeah, but you're right. We think that the model got better because we are representing more processes. But again, at the end, I will talk a little bit about uncertainty in the model and how hard to get data for this type of simulations is. But we might also be getting better answers for the wrong reasons, and that is something that we also have to think about when we run a climate model like that. But yeah, definitely, I will talk about the mycorrhizal maps in a minute. So when we now added all these missing feedback processes that the authors that wrote the reply for that original paper said that they were missing, and that's why we should not trust the original predictions that the land surface will turn into a source by the end of the century. When we rerun now with all these processes, we get a result that is actually worse than the original prediction. And so when we add the nitrogen, we have a decline of 20% in global NPP. And when we add phosphorus on top of that, we get a global reduction in NPP of 50%. But again, you guys are very interested in uncertainty, and you're right to be. I mean, these models are quite uncertain, and there's uncertainties in many fronts of this. And I will talk about uncertainty now. So what are some of the sources of uncertainty in doing something like that? So I would say that we have 3 main sources of uncertainty: a lack of process understanding. Well, we have a few uptake pathways, but we are far from having all of them. For example, we are just representing 2 types of mycorrhizal associations. There are other types of mycorrhizal associations as well. There's a lack of data. I mean, all the cost parameters were fitted for very limited sites on Earth. And so how is scaled, how can we be sure that the upscaling of this data is right as well? And then there's the lack of metrics for model evaluation. For example, we when we did the validation exercise, we were looking at variables that were readily available, like global gross primary productivity. But what about the mycorrhizal uptake maps? How can we be sure that those maps are right or not? Are people actually measuring those things so we can compare? So I will go through each and every one of these a little more specifically and give examples of some sort of uncertainties that we encounter in this. So let's go to the lack of process understanding. So how can we solve some of this lack of process understanding? Well, we could try to think about more targeted experiments, actually asking questions about the process, the cost parameters. Instead of looking at the global nitrogen uptake or phosphorus uptake, try to measure each and every uptake separately. And also identification of large scale drivers and patterns. And I mean, what are some of these large scale drivers and patterns? So one of the uncertainties, and probably the first one that we encounter here, was like, okay, we know, for example, when we fit the cost parameters for in situ measurements, we know the percentage of ectomycorrhizae versus arbuscular mycorrhizae for that particular site. But how will we know that globally? Well, there's this consensus. I think it was a paper published in 1991 showing the first global map of mycorrhizal spatial distribution. And so there's this general consensus that the extratropics are mostly dominated by ectomycorrhizae, while the tropics are mostly dominated by arbuscular mycorrhizae. But we had to give to the Earth system model the exact position of these 2 mycorrhizal types. And so how we did that was to associate one type of mycorrhizae or the other with plant functional types. And I don't know if you're familiar with plant functional types, with the concept of plant functional types, but plant functional types is the way that climate models represent species composition within a grid cell of the model. And so we have here an example. So we have a model grid cell, and this grid cell is divided into different areas. And these different numbers represent different plant species. And what are some of these plant species? Well, for a biologist, that doesn't, for a botanist, that doesn't mean much really. But for us, for climate scientists, it's kind of the way we categorize or we see plants from space. And so we can say that if the plant is a needle leaf versus a broad leaf, so information about the shape of the leaves. We can say if this canopy, this area is evergreen, so it's green all year long, or is deciduous. We can talk about the position of that particular PFT, plant functional type, on Earth. So we can see if it's temperate, boreal, or tropical. And we can also assume, we can say that if it's a tree, if it's a forest, or if it's a shrubland, or if it's a grassland. And so by associating, by having that concept that, well, we know that it is a kind of consensus that ectomycorrhizae is mostly in the extratropics. So let's give 100% to all the plant functional types that are mostly in the extratropics, like, for example, a needle leaf, evergreen, boreal, tree forest. And so when we plot this spreadsheet into the global map, we get a result that looks like this. So we have here the ectomycorrhizal tree basal area in percentage. Red areas represent the location with most presence of ectomycorrhizae, and blue areas the opposite, arbuscular mycorrhizae. And so by doing this, we were able to replicate that consensus that the tropics are mostly dominated by arbuscular mycorrhizae, and the extratropics are mostly dominated by ECM. And so it was very interesting because when we did that, the same year that I started my postdoc at JPL, 3 different publications came out showing global maps of mycorrhizal distribution. And so the interesting thing is that they used different data sets and different mechanisms, different methodologies to upscale those data sets. And so here is our map using the plant functional type concept. And here, map B, C, and D are the other global mycorrhizal maps that were published. And in this paper here that we published last year, we kind of took the standard deviation of these 4 maps. And what we found was that first, uncertainties in mycorrhizal distributions are absolutely non-trivial. And the most up-to-date estimates disagree in up to 50% over 40% of the land area. So these maps, so these yellow areas here represents the hotspots of uncertainty. And so we are not very sure about how mycorrhizae is distributed globally. And so what can we do about that? How could we eventually decrease that uncertainty? Well, now we live in a very exciting era, where we can actually have very powerful sensors orbiting Earth in a very high temporal and spatial coverage. And so when we compare, for example, what a flux tower site can measure, and a flux tower is just a tower with a sonic anemometer and some infrared analyzers on top of that. And we can measure fluxes of water, carbon, methane, radiation, and we can have an idea of how much carbon is going in and out of the system. But now we can do the same thing with satellite data. So solar induced fluorescence is a very, it's a variable that relates very well with tree productivity, photosynthesis. We can get also hyperspectral data that contains information about leaf pigments that we could eventually associate with nutrient content. And so the idea is that now that we have remote sensing, we have more data. But we need also in situ plots to understand how this data that we see from space associates with data that is measured on the ground. And also exploring links between above and below ground processes, because satellite data sees the radiation that is being reflected from above the ground. So what about everything that is happening in the rhizosphere and we cannot see with satellite data? Well, I will show here a result that we can actually kind of see what is happening below ground with satellite data. So here is the map that shows the hotspots of uncertainty mycorrhizae association, right? So one exercise that a colleague of mine, Dan Souza, that was also JPL postdoc, now it's a professor at UC San Diego, did last year was to get a map over Harvard Forest. And Harvard Forest is a fantastic place to do science because it's pretty much an open lab. So here is a plot of Harvard Forest, and the yellow dots are the trees associated with arbuscular mycorrhizae, and the blue dots are the trees associated with ectomycorrhizae. And we flew a hyperspectral sensor over this forest, and when we take the average reflectance of these 2 groups of trees, we get differences in the hyperspectral signature between those trees, among those trees. And the hyperspectral data contains a lot of information about leaf health, leaf pigments, and nutrients that are in these leaves. And so by associating different parts of the spectra with different leaf pigments, we were able to create a model that, and here's the data, the actual data as seen by the satellite, and here's the model. So we can see that we are kind of able to, of course, with some uncertainty, to determine where on Earth are which plants are associated with one versus the other type of mycorrhizae. So
Can I ask a question here, Renata?
Sure.
Hi, also the feedback was me. I have camera issues. Anyway, so okay. Sorry. When you say the associations that you have here in Harvard Forest, did someone go dig things up and look? Is that how? Okay.
Yeah. Correct.
And how many? Maybe you already said this, but how many sites are there where that's been done in high resolution?
Well, yeah, very few. So like Harvard Forest is this, they have this flux tower data running since 1991. I think it belongs to Harvard University for many years. So you know, people have been doing research for lots of, you know, many years, but we don't have many. I went to the Morton Arboretum in Chicago, and I know they also do that there. And so we have this open labs, let's say, that have this type of data sets. And when Dan wrote the paper, he showed the example of Harvard Forest, because Harvard Forest, you know, we know every single tree, how every single tree associates with mycorrhizae there, but there are a few other sites on Earth. But definitely that is something that we should start thinking about getting. This is a data that is really important to train this type of models, right? And so, yeah, definitely something that we have to start writing more proposals about that. Yeah.
Cool, thanks.
Yeah, no worries. Yeah, so I think that is fascinating, because now JPL is, you know, NASA is working on SBG, which is going to be a hyperspectral satellite that is going to orbit Earth in a very high spatial and temporal resolution. So in theory, we would be able to do this type of exercises for the entire globe very soon. So let's talk now about lack of data. So what do we have about lack of data? Well, satellite data, you know, are great, but again, we have to make the bridge. We have to connect the reflectance with what we see on the ground. And so there are a few examples of scattered data there. So like a research group collected that data 3 years ago, and it's stored in a you know, somewhere in a computer that was forgotten. And that is kind of hard when we do science, because compilation of data sets is something that extremely crucial for us in the remote sensing community, because not only because of the metadata, so the organization of those files, but also the availability of the data. And we have some good examples. I mean, the TRI database has a lot of information about leaf pigments, leaf nitrogen and phosphorus concentration. We have FRED now, which has information about roots. And also thinking about how do we link the observables and model, etc. So again, I think like this question was very pertinent. Like one of the papers that plotted the first map of mycorrhizae association was the Brian Steininger, that was a postdoc at Stanford, did, and they got this 1.1 million forest inventory plots, and using random forest algorithms to upscale these data points to a global map. But when we look at this map, where those 1.1 million forest inventory plots are, we see that they are over North America and Europe. And so how can we be sure that the random forest will work appropriately over the tropics, which are, have so limited data. And I mean, this is the example of mycorrhizae, but we can think about lots of science questions that have the same problem, right? Science is done where the money is, where we invest in science. So of course we have a lack of observables elsewhere in the world. And so how can we solve that then? Well, there's some model data fusion that we could do to reduce parametric uncertainty. And here is an example of cardamom, where we could use a combination of satellite data with process-based models to kind of estimate these cost parameters as well. And here is a wish list for experimentalists. So someone asked about the type of data that we need for the fun model to work. And this is the type of data that we need. So we need to know, so in the model, we have this separation between ammonium and nitrate. And so it would be amazing if we have this type of data. So how much would cost for buscular mycorrhizae to acquire ammonium in terms of carbon? And so, and this list goes on, right? But we don't have this type of data. So that's why we have to use the universal observables to derive those parameters. And finally, just to finish here, the lack of metrics and this actual exchange between modelers and experimentalists. I think this is something that goes on in science, where science is hard and we are so focused in our own projects. So if you're an experimentalist, you're already doing a lot of work in the lab, in the field, trying to understand your data. And if you're a modeler, you're already like, coding up and trying to understand your model. But there's a missing link between these two groups usually. And so this is an experimentalist and a modeler in the field. I went to, I got it was a kind of like not a scholarship, but some sort of funding to actually visit the Morton Arboretum and to start thinking about the uncertainties associated with carbon exudation, because something that the fun model does is to assume the resource optimization. So, well, plants will choose always the cheapest pathway, but do we have any data to actually inform that? So when I went to the Morton, we were actually measuring carbon exudates and actually thinking about, instead of just going for the assumption that plants will choose the cheapest pathway, we would actually measure how much plants are spending to acquire that. And so instead, this is how the fun model works. We can measure 5 root biomass. We can dig roots, cut the roots, dry them up, and weight them. We can get that. And we can also get soil nutrient. We can dig a hole and measure how much nutrient is available in the soil. And that's how we calculate here the cost associated with that uptake pathway. But can we actually now measure the cost? And actually, yes, we can do that. So here is us digging a fine root very carefully to not break it and insert it in a vessel. And what we do then is to pump this solution into the vessel. It's a carbon-free solution. And then we close it, put the soil back up, leave it there for 24 hours, and then go back to the field, collect the vessel, take to the lab, measure how much carbon is in the solution, and we will know the flux. So how much carbon exudation, how much carbon was exudated in 24 hours. So by having this data, we now can start thinking about actually relating those variables instead of just assuming things. So I will finish here saying that we need to start thinking about this new generation of scientists. And these scientists can call themselves Earth system scientists if they wish. Of course, we always have our preferred area, but start thinking about the Earth system as this thing that is really interrelated. And so the fact that climate change seems to be more related to climate scientists and meteorologists, and not with root folks, is somehow false, because roots do control how much carbon is going to stay up there. And so this new generation of interdisciplinary scientists need to combine theory, modeling, observations, and data analysis. And I think we here at Caltech and JPL, we are very well suited to do that. I think, I don't know if you guys are familiar with the CLIMA model, but CLIMA is this newer system model being developed at Caltech, where we are actually, instead of just prescribing everything in the model, we are actually using data to inform the model. And so we hopefully decrease uncertainty and build trust in climate predictions. So thank you very much for this opportunity. Here's my email and my Twitter. If you have any questions, comments, wish to collaborate, wish to understand a little more about the model and how to use your data or your process base that you found on the lab with the model, I will be very happy to do so. Thanks.
Yeah, thanks so much. So we have time for a couple questions. Yeah, just feel free to unmute and speak if you have a question. I have a question as well, but I'll open up the floor first if others.
I have a maybe not such a quick one. So I'm curious, like philosophically, why you feel like it's important to do this process resolved model. It seems to me like at a basic level, what you need to account for is N and P limitation is kind of the major claim that you're making. And so have I correctly understood that saying like, oh, this is the map of the ectomycorrhizae and the arbuscular mycorrhizae and some other bacterial symbioses and et cetera, that's a way of essentially connecting the limitation to some maps that you have from the literature, and not like a deep philosophical claim that like we actually need to know the underlying processes by which they acquire nitrogen and phosphorus in order to know like what the limits on carbon fixation are. Was that clear? Like, do I need to know like which kinds of fungi, right? Like, are those cost parameters very different from each other? Could I have a model where I just stop differentiating?
Yeah, so when we calculate the cost parameters for each uptake pathway, we need to know now. So now we know when the model was fitted, we know how much carbon a plant would invest via that uptake pathway, let's suppose arbuscular, if that amount of nutrient is available in my soil and I have that amount of roots in my soil. But I still need to know what is the mycorrhizae in that place, right? Because so it is less philosophical and more practical. It's kind of well, we need to know if that place has what type of mycorrhizae is there.
No, but why do you need to know that? That's the part that I think is like, wouldn't it be better from the modeler's perspective to not have to know that?
Well, but we have a explicit representation of these two uptake pathways. So for example, let's suppose that we are in a place where there's no ectomycorrhizae. We are in the middle of the Amazon forest. Why would we include that uptake pathway in our calculation of resource optimization if there's no ectomycorrhizae there? You see what I mean?
I do see what you mean. I'm saying like, I'm imagining the simplest possible model, which is that plants invest carbon to get nitrogen and plants invest carbon to get phosphorus. And you have one parameter for the cost of each of those things, right? Like you've convinced me that I need to account for those things. But you've shown me that if you put data in that says where the mycorrhizae are, that like the data tells, informs the model in some way on its output. But do I need to differentiate between them at all? Like, have you tried just using a simpler representation of the set of processes involved?
But I think that goes back to the first question, right? When those guys published the paper in 2015 in Nature Geoscience, and a reply came back immediately afterward, those guys use a very simple model. They use bookkeeping methods saying, well, this is the amount of nitrogen and phosphorus that we need to acquire to support the regional photosynthesis, without no information about which feedback process, which uptake pathway. And people were like, well, your assumption is wrong. You're not representing lots of important processes that are not being considered in your model. So I see your point, like sometimes it is better to have a simpler model to get a simpler question. But this question is not simple. You know, nitrogen phosphorus acquisition is not a simple question. And so we need a slightly more powerful tool to address that. And that's why we need that extra information.
But I'm not
and
sure. I don't mean to press on this, but I understand what you're saying, but I don't think you've made the argument, at least in the talk, that the more complex model is necessary. Maybe you did do it in the paper. I haven't read the paper in a while.
I think, well, at the beginning, I wanted to show this slide showing about the recycling and the misting processes. There's something in climate change that is called acclimation, right? So if you, let's suppose that you're simply assuming via bookkeeping methods, right? You know how much phosphorus is in your soil and nitrogen, and you know how much phosphorus is required to support that photosynthesis, and then you calculate things. That works now, right? That's something that works now. But climate is changing, and when climate change, things change. And so that's why you need a process-based model to follow the change. So what now we are informing the model is the process and not simply a cost that might change in the future, right? So because we have more CO2 in the atmosphere, plants will photosynthesize more and the cost might change. And so if you don't represent that, you're missing out. You're not getting the full picture of climate change.
Okay. I'm going to email you. I'd like to talk more, but maybe
sure.
I'll let John ask his question.
Okay. Thank
Sure.
you.
My question is kind of related in the sense of when you are choosing to break down these distinct processes as kind of individual entities within the model, is the justification for what divisions to make and which resolution to make those divisions at, do those typically tend to come from the known biology, or do they tend to come from kind of just empirically, like, oh, it gets a little better if we kind of make this distinction, like AM versus EM, you kind of are saying like all mycorrhizal are like the one or the other category, the two categories behave differently, but within the category, there isn't much that we care about. Is that like informed kind of like what's known from the mycorrhizal scientists doing the biology, or is it kind of like there's still more that we can learn there?
Yeah, no, that's an interesting question. I added lots of extra slides because I hoped people would ask questions about that. Hopefully I add that one. Oh yeah, here. So I talk a little bit about the development of the fund model, right? And so when the original fund model was built, they didn't have all these uptake pathways. They have something called active, which was mycorrhizae, let's say, and bacteria, and something called retranslocation. And when they fit the model to the data, they get a reasonable agreement. And so along the years, Eddie was a postdoc of Rich Phillips' postdoc in Indiana. And Rich is a biologist. And so they were really thinking about how to add information about mycorrhizal uptake in a model like that. And what they found out was when they started adding new uptake pathways with new cost functions, the agreement between observation and model start to increase. And so, yeah, there is a biological there is a biological need, or like for process understanding, an increment of process understanding with uptake pathways.
Yeah, that's great. And just to follow up on that, I really loved your slide about like what are the known, unknown parameters and variables in the model that kind of need to be measured. But do you have like a similar sense of known unknowns for process level phenomenon, such as this kind of distinction of like active splitting into AM, EM, or is it mostly like unknown unknowns from that point of view?
So I published the paper about the mycorrhizae spatial distribution last year, and this year I published a paper in James just talking about the results of fun in phosphorus. And that paper was really hard to publish because the reviewers really wanted the future development of the model, and rightly so, right? Like what's next in the fun model world. And so the final session of the paper, I talk about, it's kind of a philosophical, it was really fun to write, but we talk about where to go with this. And you're right, there are unknowns, right? But there are also known unknowns. Like, for example, we know that plants are very good in changing stoichiometry, right? So plants may adapt how much phosphorus and nitrogen they may use to support photosynthesis. And we can see like a general trend of leaf phosphorus and nitrogen going down with time. This is measured, this is observed. So it's like plants somehow are investing less nutrients to support the same photosynthesis. So they are adapting, right? They are changing with climate change. So one thing that we might need to add in the future is this adaptation of plant stoichiometry as well to account for this type of questions. And other things, like in the model, for example, we just retranslocate nutrients from leaves, but we know that retranslocation can also happen from fine roots, for example. So we might want to add retranslocation from fine roots. Anyway, there are lots of space to improve this model and other processes that we could consider here. But yeah, it's exciting because it's definitely not a solved problem. It's definitely something that we have to keep thinking about.
Great, thank you.
I have one, I guess, last question. I know we've got 2 minutes, but sort of a simple question. I realize we've been saying this in various ways the whole time, but I still think it's maybe worth asking again. How much from the ground up, sitting with mycologists in this case and just saying, what do you think is like, what are the 5 most important things from mycology? Like how much of that is happening? Because I think what several people are kind of touching on here in different ways is a question of how have we decided what should be important, and is it overly driven by things that we can measure? Like if I ask a mycologist what type of plant associates with EM and AM, what would they say?
Well, I don't know. No, I don't know if I get the full question. Like you're talking about the spatial distribution or like the importance of considering.
So you've said that EM and AM associations are a big thing, right? And the problem is we don't actually like what's this. You may not know this, but I'm just curious. Like with the state of knowledge, it's just as an example, the state of knowledge on what the drivers are, but like what determines which type of association is formed.
Oh, like yeah, I don't know actually, like, but that's not.
Yeah, well, and and is there a reason that it has to be associated with something that's easily observable?
Right. But I think like for example, when Rich and Eddie, they suggested adding this differentiation between these 2 types of mycorrhizae, it's because people have been talking a lot that mycorrhizae is extremely fundamental for nutrient acquisition, right? There are some estimates, I think there's a paper from a group in the Netherlands estimated that 80% of all the nutrients come from mycorrhizae association. So they were like, well, we need to differentiate between these 2 in order to get a better agreement with the data that we have. And they have, again, these are all observations. These are sites that the data was fitted for, and they have like observed retranslocation. So what they found was then when they added these new pathways with new costs, they were able to get a better agreement. So I think, like, I don't know if I understood your question properly, but when Josh is a climate scientist, and he developed the concept of the fun model to kind of account for the cost of carbon to acquire nutrients. But when Rich joined the project, Rich was like, that doesn't, that's not really important to me as a biologist. Like, I really want to understand how much nutrients this or that type of mycorrhizae uptake, and how is the feedback process is in there. And so that is the consideration. But I think it goes back to the question of what are some of the missing processes. I mean, we're dividing these two groups in a very general way, right? We're saying, well, AM behaves this way all around, and is different from ECM that behaves this way all around. And that's not necessarily true. I mean, there are thousands of different species of AM and ECM, and each and every one of them might have a slightly different cost benefit for carbon acquisition. So it was really to try to understand this, because mycologists think about mycorrhizae in these two ways, right? When I was in the Mortimer Observatory and we were actually measuring carbon exudation, we would go to a plot and they would say, oh, this is a needle leaf plot with AM, and we know that. So they were not very interested on the specific AM, but they know those trees were associated with AM. And so that's why they went to there and measured the exudation there. So now we could have this data set that tells us, well, a needle leaf associated with AM is exudating this much carbon, versus a broadleaf that's associated with ECM it's exudating that amount of carbon. So
right, but
yeah.
It doesn't tell you that the actual important thing there is the EM versus ECM, right?
No, but again, I think this project in particular is thinking about the feedback to climate, right? So carbon absorption. But no, it's not telling any information about the intrinsic differentiation between these two pathways. No.
I think, yeah, we're over, so I'll let it go. But that's sort of my question, is less about the different types, but what the fundamental divide is, and whether we're building from a knowledge base of that or how things are.
Yeah,
anyway.
that would be fantastic. Yeah, that would be great.